Thank you very much for having me for this. This is really a wonderful opportunity to come talk about science and share what I've learned from being at the public interface and talking about a very important topic in science that has really been a subject of discussion in all of our family and all of our friends, many of our circles. And what I would like to do today is give you an idea of we'll talk about some of the facts around genetic engineering, we'll talk about the relative risks and benefits, and then my most important part is how you, as people who are concerned enough to come out on a nice cool night to come do this, how can you better engage family and friends? My most exciting part of tonight is we will have time afterwards for your questions, and so that's where I'd like to spend most of the time. We think about Charles Darwin, and Charles Darwin was an observer. He made many excellent observations by looking at animals and plants around the world and the differences that he saw between them. And when you look across plants and you look across animals and you look across this room and you see the beautiful diversity that we see, all of the differences that happen between us happen because of changes in genes, that the genes are different from one person to the next, from one plant to the next. And this is what gives us the very basis of variety. And this was Darwin made all his observations years before we knew what genes were, years before we knew what DNA was. And Charles Darwin, what would he be doing if he was alive right now? Probably be clawing at the inside of his coffin desperately, but if he was alive right now and here with us, what would he say about the science? And I think he would be amazed at how our ability to look into the cell and into the DNA and into the DNA sequences that are the blueprints for our lives, how it really mirrored very well what observations he made in the field with his excellent science. So this is a little bit of a tribute to Charles Darwin, that we just can't now describe what's there. We have the opportunity to change what's there. And what I'd like to explore with you today are some of the ways we do that, some of the really great technologies that are coming, and some of the ways that maybe we can think together on this to use technologies in wise ways that satisfy our most important values. So, as was mentioned before, I'm a researcher first, and my real love is in the lab, and I'm an administrator at a university, and that's okay. The service side is good, but my real heart is really in the laboratory, and I service the state of Florida where we have many challenges to agriculture. We have a citrus crop that's dying. We have tomatoes and blueberries that have threats. All kinds of problems with labor and in competition. It's really difficult in agriculture right now. And so we're using every tool we can to solve the problems that face our farmers and that produce good food for the rest of our country during the off time of the year. We are not what you would call a GMO state. We don't grow any crops that are of the genetically engineered variety. Very little, maybe a little bit of cotton, a little bit of soy, and a small amount of corn. But for the most part, we're growing tomatoes, strawberries, blueberries, peppers, beans, citrus, things that are not genetically modified, at least through the genetic engineering methods. So we'll talk a little bit about that today. The other big thing that we do, what my lab does, is we study how we make fruits and vegetables taste better and what are the genes that control the flavors. How do we use light in clever ways to control the way food spoils, to control the nutrient value, and to control how good it tastes. These are all things we're able to do with non-chemical treatments of light, and that's really, really neat stories you'll hear a lot about in the next couple of years. And we're doing some really cool stuff with drug design. A really novel method that I just adore that hopefully Darwin on the Palouse 2019, I'll come back and tell you about our potential cure for MRSA. We got a lot of neat stuff happening in the lab. So all of my funding and reimbursement is online. I'm super transparent about all that stuff. You can go back and poke through how much I get for a plane ticket to go to a conference and who picks up the cab fare and all that stuff. It's boring, but you can see it. And all the slides that I ever provide are there for you to use. So if you'd like to take any of the slides you see today and use in talks with your family or friends or your church group or whatever, feel free to take them. I'm more than happy to provide that, and let me know if I can ever be of service in that. So what I need you to take home, I want you to understand what genetic engineering is and what are the traits that we currently use, what is it not? I really want you to understand the nuts and bolts of this technology. And I'll show you why it's an important tool, and I'll introduce you to some of our new tools. And this new tool called gene editing or genome editing, amazingly powerful tool. And this is something that will be revolutionary in agriculture and revolutionary in medicine. And there are a number of children alive today who had their leukemia basically cured by this kind of genetic tweak. And an amazing story, we'll talk about that too. But most of all, I really want to deputize you. I would like you to have the confidence to be able to talk about these topics with family and friends, and not just genetic engineering, but any controversial topic. In the world we live in right now, where it's really hard to tell who to trust, and it's really hard to tell what's the truth. When we talk about climate, vaccines, evolution, did man land on the moon, right? There's all these topics where there are pockets of people who have curious skepticism about it, which is maybe healthy in a way. But how do we communicate the science and what are the facts? And how do we talk to people who have different opinions and work very well to change their mind? And that's what I'd like to wrap up with today. So, this is a gray wolf. Gray wolf was here on the planet along with human beings, and some of these gray wolves found that by following human tribes around, they could get some benefit. Humans that were followed by gray wolves got some protection. They had a really angry animal around that kept other rodents out of their food and maybe protected them and gave them an alarm if other people were approaching hostilely. These animals came into the human provision in that we changed them over time. Humans selected different traits inside these animals to develop a wide variety of different species that are genetic modifications of that original species. Really, when you think about it, these guys are all GMOs, right? They're genetic modifications of that original species, all propagated by humans and selected by humans to do what humans wanted them to do. We selected traits based upon human necessity. And the same thing can be said about other food items, things like chicken. So, well, these are chickens that were from broilers from 1957, a number of days after they were born, and compare that to 2001, and you can see the tremendous difference that just better genetics gives us in terms of our food. That larger animals with more flesh, less body cavity, at the same number of days after hatching. And this is another testament to humans manipulating genetics. Not GMO per se, but changing genetics by improving the organism through breeding. Matching the best with the best to get something better. And this is another way in which humans have always manipulated genes. This is a really cool slide. And this shows the genetic origins of all of the food crops in your grocery store. And what's really exciting about this slide is that it shows us that all of the things we consume really come from someplace else, and that they were brought from where they belonged and from where nature put them, and in the ecosystems and the environments that they served, to places where they don't belong and where they actually compete with the natural flora. So when you look across this entire scope and you see very few things come from North America, maybe you got blueberries and huckleberries and things up there. But for the most part, things that we're consuming are aliens on our soil. They're plants that belong somewhere else and were brought here and genetically improved because we decided that they're important to feed us. And this is a really important slide too. It really leads into this one. That humans have made tremendous genetic changes to the plants that we eat. The genetic changes to that plant on the left, that's teosinte. And teosinte is a branched, weedy, leafy plant that grows in Mexico. And it makes a little stick with about 13 little kernels or 10 little kernels. That's the great-great-grandparent of corn. Over here on the right is tomatoes, which grow on the ground, on vines, on little tiny capsules that don't taste very good. Some of the chemicals that are in there are the same components of cat urine that are present in wild tomatoes. But through many years of breeding and eating cat pee salsa, humans improved tomatoes and corn to their modern day varieties. And all of this change occurred because humans took a place in changing those genetics to make something that was better for us. And so, you can see kind of where I'm going. To me as a scientist, changing genetics doesn't get me too nervous. It's something that we've been doing in very random and in very wild ways for a long time, creating changes that couldn't happen because of nature to better suit what humans wanted. And really that's the prelude to what we'll talk about with genetic engineering, where now we're able to, because we understand what genes are and how they affect traits, how we can move them in a custom way. one by one, to change traits that we want changed. And I'll show you how that works. We have to start with a little bit of a biology lesson, and a lot of this is common for some of you, but let's go over this anyway and use the analogy of your home computer. What we call the central dogma of molecular biology is the fundamental path by which information flows through a plant or through a cell. And the way you think about this, and let's start over with your home computer. You have a hard drive that's sitting inside the box on your desk. You never see it, right? It's always in there unless something bad happens, but it's always in there. And it stores all the information. It's the ultimate source of all the programs for that cell. You have this thing called a USB drive. It's temporary. It's transient. You can take the information from that hard drive and take it somewhere else. And a lot of times it ends up in the lint trap of your dryer. I find at least one in the bottom of my washer every week. I mean, it's a temporary storage solution for data, right? And then sometimes you can take that data to a place and, like, say, take it to work where I can get a nice color print for free and print that out and get a product that comes from the information from the hard drive of my home computer now coming out at another place. It's a lot like that in the cell, where you have DNA, which is there's the chromosomes, it's the hard information storage, the blueprint of the blueprint of an organism locked away at the center nucleus of that cell. This kind of temporary form, this RNA molecule, takes the information from the DNA in a copied form and goes out into the cell, where then some machinery takes that information and creates a protein. And proteins are the enzymes, they're the structural parts of cells. So this is how the information in DNA gets really translated into a final product that is functioning inside the cell. It's actually a very well-studied process. We understand it very well. So if you wanted to make a genetic engineering change, what do you do? And there's a couple ways that we can do that. You can first put new information on the hard drive. You could wipe a little piece of information off the hard drive, or maybe you could destroy the USB drive. And that's exactly how we do genetic engineering in the cell, adding or subtracting things from the nucleus or destructing that RNA intermediate. And I'll talk a little bit about that. It basically is this process. You can add something to the hard drive, delete something from the hard drive, or just block that information in between. And I'll show you some ways that we do that and some products that are present and products that are coming. It's actually a lot simpler right now than you'd be surprised how simple these things are. The way that that's been done in the past. So how do you change a hard drive, information on a hard drive, if you don't know what genes are and you don't know what gene sequence is? How did we do this back in the 1800s, in the early 1900s, in the 50s and 60s and 70s, 80s? We did it this way. We made crosses, mixing genetic information between two plants that may never cross naturally, are able to cross them because we're really good at doing that as plant biologists. We can create crosses that wouldn't exist in nature. Take a plant from Mexico and cross it with something from China. Never would happen in nature, but it's just something that we can do to capture the genetic diversity in one place, maybe make something a little better. In the 1940s and 50s, you could use X-rays, well, still do, but back then started using X-rays, started using chemicals to damage DNA. My voice is a little gravelly. To damage DNA, because you could damage DNA in ways that you didn't know what you're doing, you just knew that you got a product that you liked. There's a lot of great examples of barley, of many different fruits and vegetables that came through that particular process. No one knows what genes were affected. You just know that you got a product that was better because the DNA was changed. And perfectly acceptable to grow them in organic production, perfectly acceptable, no problem. We don't know the changes that occurred. Nobody really cares. It's kind of neat. We also can double and triple chromosomes, and that's been done for a long time by using very toxic chemicals that are frequently used in chemotherapy, or at least were historically, to destroy the ability of chromosomes to move into two daughter cells. So that was no problem. People did it. They got better products out of it. Nobody cared. This was genetic engineering before we could move a single gene. This is what happened in the 50s and 60s. Now there's this idea of genetic engineering, which we do today, which really is just an extension of conventional plant breeding in that we can move one single gene for one single trait. And we'll talk a little bit about that. But the basic idea is, here you just randomly whacked stuff on the hard drive, and maybe you got something that came out better. Down below, we're using great precision to add one thing or take one thing off. And that's the contrast I want to leave you with there. So what we know about genetic engineering. And this is just a cover slide that covers the entire scope of things. We won't go into the details. But the techniques used pose no more risk than conventional breeding. And what that means is something very important, is that when we cross two plants together that we've never crossed before, there is a little bit of risk there. I mean, plants produce some toxic compounds. And could we be producing something that makes a new allergen just by crossing? And it has happened. So this is actually a little bit better because we have a little more control. Genetic engineering, we at least know what we're moving. In 20 years now that these technologies have been used, they haven't been related to one single case of death or injury, and that there's really just a couple of traits. We'll talk about that in a minute. And then this idea of gene editing, and I really want you to think about gene editing. I want you to use tonight as really maybe your first contact with the concept, and I really want you to think about it and study it, because this is going to change everything unless we regulate it to death, and I'm really nervous about that. So, what are the genetically engineered products? First one came out in 1983 when the gene from human insulin was placed into a bacterium. So we took something out of a human being and stuck it in bacteria, which is about as wide and freaky as you can get, and grew up big vats of bacteria and took the products out of them and injected them into our bodies. And 40 million people use insulin therapies, I think just in the United States. And it's really been a lifesaver, because before this, they had to purify insulin from the pancreases of slaughtered cattle. And so this was a huge step forward, because you could get much pure human insulin from bacteria because of recombinant DNA technology. You could also do growth hormone, which was really huge. It had the possibility to extend the long bones of people with growth diseases. And even more recently, rare diseases have been countered by drugs which have been manufactured in, say, carrot culture cells using genetically engineered cells. So there's a lot of products that have come through genetic engineering. Most of the vitamins you get, different compounds, artificial flavors, a lot of this comes from bacteria and fungi that are genetically engineered. And people don't worry too much about that. Where people start worrying about it is when we talk about crops. And I think I understand why, and we can talk about that afterwards. But right now, there's really just 10 crops which are genetically engineered. And usually an audience like this, I'll usually poll the audience, and people say wheat and tomatoes and strawberries. No, this is it. And we'll talk about them a little bit. There's really just 4 that are in your food commonly. And this is usually the cornstarch, high fructose corn syrup, soybean oil, soy solid, sugar from sugar beets, or canola oil. It's just the products that are in processed food. 90%, or I should say 80%, of your corn and soy are going to animal feed. Another 10% are being used as fuel. And so, most of this is not even intended for human use directly. So there's really just those 4 and little dusting of ingredients in your products. The other things are used for animal feed. Okay, so you have alfalfa there, and then some cottonseed. Cotton, obviously, for fiber. Squash and papaya, very limited acreage of squash, very, very limited acreage of Hawaiian papaya. And now this year, apple and potato will be new on the market. You should see a non-browning apple and a non-browning potato available this year for the first time. So these are new ones. So this is really our only suite of crops that we're talking about when we're talking about genetically engineered crops. Maybe a little bit smaller than what you expected. What about the traits that have been engineered in? And we can talk about the traits in detail afterwards if you like, but I'll just give you an idea on those. The one point here is that the main benefit has been to egg producers. And that's the one big problem with this, is that the companies that when they created these products, they didn't do a whole lot of talking about what they were doing and why they were doing it. They produced it for farmers to have better seeds and cheaper production, and they didn't worry about you and me and talking to us about our concerns. And this is where they screwed up majorly, and they know it now. It took them about 20 years to realize that they screwed up in a big, bad way. So these were all benefits to the producers, and not producers for folks like you and me necessarily that we saw as direct benefits of these technologies. And that's part of the reason why there's been so much discussion in negative ways about the technologies. But what are the traits that are encoded? And the traits are a couple different ones. And this is an important one. Cotton and corn are made to be resistant to insects. The plants are engineered to contain a protein from a bacterium that is toxic to certain caterpillars. So in other words, if you're a kind of organism that feeds on corn, this protein that's in the corn will suppress your digestion and you die. And that's only in that particular organism that feeds on the corn. It's perfectly safe for you and me. It's kind of like how you would never feed chocolate to your dog, but you'll eat it all day. You know, it's some of the compounds that are toxic to one organism are not toxic to others. And so this is what, that's how this one works. Very important, because when you have the corn protecting itself from the organisms that feed on it, you don't have to have the plane fly over and spray the entire field. And so you've seen insecticide use for broad spectrum insecticides decrease tremendously on corn and cotton in many places throughout the United States, between 50 and 90%, depending upon where you live and the insect pressures. So this is a very important trait. The other trait is herbicide tolerance, and this is one that is really important, but and has a lot of criticism associated with it. But what you're looking at here are the crops are the dark green and the weeds are kind of the yellow. And that was a day after treatment with an herbicide called glyphosate, which is a compound that binds to a specific enzyme in the plant and stops its metabolism. That's toxic to just about all plants. It's a broad spectrum herbicide, and it interferes with an enzyme that we don't have. It's not a human enzyme. It's an enzyme that's only in plants to make an amino acid. We can talk about the details of that later. But the basic idea is that you can engineer the plant with a form of the enzyme that is not sensitive to glyphosate. It comes from bacteria. So you take the gene from bacteria, put it into the plant, and now the plant can grow in the presence of the chemical. And so what you're looking at here is a farmer planted the seeds, let everything grow, weeds and plants, and then goes over and sprays this chemical that kills only the plants that are sensitive to it. The plants that are engineered survive. And it allows farmers to grow plants without tilling. I neglected to talk about with insect resistance, but we can talk about these two together. So here you have, I've discussed the upsides of this, right? You don't have to use insecticides, and you can use a very low toxicity herbicide that is cheap, and you don't have to use tilling. But what are the downsides? Here, well, let's talk about it. So the problem is when you have a solution that you use over and over again, you develop resistance. And this is a huge problem. There are places where the insects are becoming resistant, and this doesn't work anymore. And now you have to call up the spray plane to come hit your field. The herbicide resistance. There are weeds that grow right through the herbicide, and they don't care. Darwin would have picked this out, right? Darwin would have said this is a bad strategy. He'd just go ahead with one herbicide over and over again. You're going to select with a very strong selection pressure for the weeds that can survive it. They evolve around it. This is evolution in action. And so on Darwin Day, it's important for us to take a good look at, here's two good solutions that worked well for a long time that are losing their edge because we didn't plan correctly. And those of us who saw this back in the 1990s said there is no way that this resistance will last 10 years. It lasted 20. But you still see a lot of places where this is a very important problem in farming. The virus resistance is in papaya and squash. And virus resistance looks really good. It's a mechanism that's so robust and very difficult for the virus to get around. The advantage here is that you don't have to spray for the insects that transmit the virus, which is currently the way that you would control it. And so this is a very, very important trait that saved the papaya industry in Hawaii back in the 1990s. That's it. Those are the only three traits. It's herbicide resistance and at least the major traits. We have some minor ones here and there, but insect resistance, herbicide tolerance, and virus resistance, and just a couple of crops. And most of the things that have been done and most of the solutions we have never been implemented. And we'll talk about that next. That's kind of the sad part for me as a scientist. So what I showed you here are the ways that big companies can create plants that have a couple of different traits that are good for farmers. They didn't roll them out very effectively. They didn't talk to us much about it when they did it. And now they're surprised that people are a little bit curious and suspicious, think that there's a problem. Now, for me as a scientist who studied this my entire life, seriously, I studied this since I was 10 years old. I took out my first book on recombinant DNA technology, a nerd. And I did that back in 1977. I read about this stuff. I thought it was so cool. As a scientist at a public institution, one of the things I really think about are how do we take care of the environment? How do we take care of the needy who don't have anything? How do we come up with solutions for people in desperate parts of the world? How do we help the American farmer farm more effectively? And how do we have better products for consumers? So what I'd like to do now is walk you through a few of the things that we have created with your tax dollars that are not being used. We know that we can create versions of plants that are biofortified. And what you're looking at here are 3 different food staples in rice, cassava, and banana. On the left in their native form, and on the right in their vitamin A enhanced form. So they contain beta-carotene, the stuff in carrots. It's very easy to move the enzymes that create beta-carotene from one plant to another. And in this case, they've been placed in the plants where they normally wouldn't be found to make them more nutritious for those who consume them. And vitamin A deficiency is the leading cause of blindness in many places in the world. So this is a very important trait. Disease resistance. So there's cases where I talked about papaya, but cassava, another world staple that has viral problems and could be alleviated with this technology, has been, just not deployed. We have a disease in Florida, citrus greening disease, that we have trees that are genetically engineered now for 7 years, no sign of the disease, yet we can't grow them. Bananas, xanthomonas wilt, breaks my heart to see it. In Uganda, there are many trees on smallholder farms where people grow a cooking banana, they call matoke. The trees are very sick and die in their small groves, yet behind the barbed wire and the fence are perfectly healthy trees that they can't access. And they're genetically engineered, hopefully will be out and available soon to resist the bacterial wilt. So there's ways that we can solve problems with diseases. There's ways that we can suppress allergies in things like peanuts or wheat. Very important here in the industrialized world. And we can also come up with ways to mitigate the effects of changing climates and wild weather, that plants that can resist weather extremes, floods and drought and heat and cold. Very important. And everything I just showed you in the last 4 slides have a couple things in common. They're not big products by multinational companies. They're products you paid for, or small governments around the world paid for, maybe Gates Foundation, things like that. But they're products that have been created and are not being used in part because of a negative public perception. And if there's one thing I'd like you to take out of here today is the idea that genetic engineering can be a tool to do some very good things, yet those good things are not yet coming to application. And then we'll talk about that and how to communicate that towards the end. There's also some animal ideas where you can create chickens that don't get bird flu and salmon that get to market size in half the time. That salmon now is 28 years old. That technology, not the salmon. Salmon's long dead. He was delicious back in 1990. You know, I'll talk about this before we go into the communications part, just because I want you to know gene editing. So we talked a little bit about traditional breeding. You cross 2 things together and you get a mixture of the 2. Genetic engineering, or this GMO technology, we take, and those just represent chromosomes with the little beads are different genes, where you can add a new bead to that string of beads to give you a new trait. And that's what we have for insect resistance or herbicide tolerance. New information on the hard drive, right? Gene editing is much different. Gene editing is a case where we can go into a cell and make a specific change in a specific gene. It's like being able to go into a library, and if genetic engineering is putting a new book on the shelf or taking a book off the shelf, gene editing is changing an A to an O in one word, in one paragraph, in one sentence of that book in that library. Tremendously powerful. And this technology will help us do some beautiful things, and I'll talk about that here. I won't go into the details just because we don't have enough time, but what are the products? And this is one of the reasons people like gene editing and don't necessarily like genetic engineering. When you make a genetically engineered product and you add a gene, you add new information to the hard drive, you add some other stuff along with it. Other sequences that move in when you add that gene. And people sometimes will say, well, this is why I object to it, because you have all this other collateral stuff that doesn't belong there. In gene editing, you can essentially get, so here's a natural mutation that would happen, say, in this gene sequence, which is A's, G's, C's, and T's, that make up the sequence of a gene. Here's a case where maybe a gamma ray from the sun, or emission of radioactivity from the Earth, or just ultraviolet rays or sloppy DNA replication made that change, which caused a new trait that may be very important, or maybe not. Gene editing, we can make that same change. So the point of gene editing is that you can create the changes you want to see and have a final product that is indiscernible from something that came through traditional breeding. That's why it's so powerful. It's being able to do, if we had 10,000 years to find that one single mutation, we could do it. But here we could just make it. And that's really why this is such a special technology. The applications so far, and this is a beautiful one. So here's, you got beef cows and dairy cows. They make milk, bad beef. They make beef, bad milk. They don't have horns, they do. If you cross the two together, you get a cow that's not good for much of either, okay? But the reason they don't grow horns is because there's a gene that controls the production of the stuff in that spot that eventually comes out as a horn. And that gene is disrupted in the black Angus. And they've known this for years, that the gene that leads to that little horn bud is disrupted. And so what if you could make that same change in the dairy cow? Could you get rid of the horns? And they did it. What they did was they took that same information and just made that same change in the dairy cow. They didn't have to exchange any genes. They didn't have to switch anything around. They just took a few words out of that book and made it what they wanted it to be. They disrupted that function of that gene. And now you get, there they are right there. This is one over here. This is what they should look like at that growth stage. And here they are over here with no horns. Why that's important is because horns are, they're dangerous for other cattle, they're dangerous for humans, and it's very unpleasant to remove them. You have to do what's called scooping, where you scoop them out of their head, or shoot them full of, whatever it is, the numbing stuff, Novocaine, and then have to burn them out. And so this is better for the animals, it's better for the farmers, it's less cost. This is a good thing that could be done. So this is a nice application of genetic engineering technology. There's a pig disease called West African swine fever virus. It's very bad in parts of Europe and devastating to swine populations. Warthogs don't get this disease, but you can't cross those. They're not interfertile. But you can take the same gene that has a mutation in the warthog and move that gene move that same sequence, make that same change in the domestic pig, and potentially confer resistance. They haven't tested it yet. They made the piglets last year. That's them over there, but they're testing them this year. So here's a way that you could take a gene that exists in nature and bring it into your domestic population in 1 year, rather than through many, many years of breeding. So please read on this. And whether you're excited about it or whether you hate it, I would really urge you to participate in the public comment period. Really important, because this is a technology where the FDA is now taking our advice and guidance, and they're looking for really precise information, scientific information. So share what you know about it. It would be really wonderful to do that. There are other cases we can talk about later about how this has been applied to medical problems. They can cure muscular dystrophy in mice. They can cure liver diseases in mice. They can use this gene editing technique to make cells that can attack cancer and not be rejected by the body. And they've cured a couple people with leukemia from this. Very, very powerful technology. Neat stuff. All right, so last thing I want to talk about, wrap it up with this. If all this is so good, why is there such a problem? And we have these wonderful innovations that are not moving to applications, innovations you've paid for, innovations I work with people who develop, and we don't get them to the people who need them. And why is this a problem? And it's a couple of reasons. One of the major reasons, and this is a good thing, is that people are asking questions about their food. And this is wonderful. To me, I am so excited about the fact that people finally care about asking what's in it, where's it from, who did it, all that stuff. I think that's great. I think it's a really wonderful thing that we're able to be at this point, because now we as scientists, farmers, concerned citizens, whatever, can now step in and give them those answers. The problem is that we've been doing this wrong. Scientists screw it up. Farmers screw it up. And for the most part, most of you, many, at least are science-minded, thinking about these things. We probably don't communicate these things very effectively. People are interested in knowing what's in their food. They want something healthy for themselves and healthy for their families. There they are right there. Moms are worried about their kids, and it's a wonderful thing. But when there's so much misinformation about this technology, people will use fear to scare them. And we see it on TV. We see websites which will sell you products, who will scare you about your food and then sell you a solution. They're everywhere. What do we do as scientists? And I did this for 12 years before I realized I was screwing it up. I come back with facts. Well, obviously you didn't see figure 5. Maybe you need to look at something with the p-value on that one. That's a little bit more important. Maybe if you looked a little bit, maybe if you look at the significance underneath this curve, we could see if we use a Mann-Whitney-Hughes test, we could transform those data. I did that for 12 years, and I didn't change anybody's hearts and mind about this really important topic. And it's what happens with scientists. We're very good at talking to each other about science, and we're very good at arguing with each other about facts and evidence. But when someone's scared about their family and their food, all the facts in the world don't matter. It doesn't matter. It's what they feel and what they believe, and in their hearts they feel there's something wrong. And in order for us to be effective at changing that, we have to be able to communicate with them in a different way. And that's the strategy I'd like to talk about. The big problem is that we have an erosion of trust that happens from people who are trying to make scientists less viable and erode the trust we have. And we see this in climate and vaccines as well. This is a major problem. We also have a lot of stuff that looks like it's facty, but it's not. A lot of information that gets out there that really scares people and becomes part of a story that really does scare those people who are trying to make decisions about their food. So the negative effects: higher costs for us, bad policy, innovation is arrested. And the big problem we have from this is that there's a lot of scientists who are just afraid to communicate with the public. Scientists are uncomfortable going out and talking to the public, and farmers too. The next part I didn't necessarily know if I wanted to talk about, but I guess I will, because I think it's important, and it shows the importance of you. It shows the importance of the skeptical movement, of the scientists getting out and using their voices, of people who just love science and want to spread and share science. It's important for you to see a little part of what happened here. So I've always done workshops and outreach. It's been a cornerstone of my program. I do workshops, I teach kids, I do stuff. I spend a lot of time in schools, talk to physicians, dietitians, and how do I pay for it? When I go somewhere to give a talk, like for an industry thing, or for corn growers, or for whatever, they say, how much do you charge for speaker fee? I say, make a donation to my program. And they put money into a university account where I can buy USB drives to hand out with information. I can put out coffee and donuts. I can stay an extra night in a hotel if it means staying after a conference or something to do the workshop. So it's just that kind of thing. None of it goes to me, zero. And it could. And we're talking a substantial amount, but I put it into this program. A company realized that I was doing this and saw I was doing a good job, and I knew somebody, an old friend in the company, said, we'd really like to make a donation to your workshop. That would be fantastic. We'll make a donation to it, and then it'll allow you to do more of these things because you're doing a great job. And I said, that's cool, that's great, but you have no control over what I'm going to talk about. They said, of course not. We just want you to do more of what you're doing. And that was good. And so not long after this, a $25,000 check went to my university. And I thought, this is the best thing ever. Because thanks, Monsanto, for allowing my university to get money to help me do this. I thought this was a great thing, right? But you can see the problem as it happens. A lot of folks don't like that I go out and talk about science. And you can tell, I'm pretty even-handed. I tell you about the strengths and weaknesses of this stuff. And some folks use the Freedom of Information Act to gather all my emails. A couple of folks have taken over 30,000 of my emails that I just give up. I don't care. You can do that. You can write for them too if you want. It's boring. But one of them talked about this donation to the program. And from that, there's the letter asking for it. But long story short, there's my inbox, 26,517 emails. I don't get to everything. Anyway, there were emails in that FOIA request that were delivered that I never even opened. And so this happened, and then this happened next. Front page of the New York Times. And the front page of the New York Times says, emails reveal academic ties in food war, industry swaps grants for lobbying clout. This author called me a lobbyist, said that I trade my academic integrity for money, that I'm paid off to lie about science. I mean, this is what you get. And I told him that none of this stuff is true, but he did it anyway. And if I paid because of this, and then we returned the money. My university said, if this is the way it's going to go, we're taking that money, we're giving it to the food pantry, and they did that. So I didn't even get to use $1 of it. But that didn't change things online. Yeah. Cool, huh? So, yeah, they let me have it, both barrels. The sad part is that they doxed my family. They broke into my office at work. People in my lab were threatened. I mean, it was a really, really bad scene, and still goes on today. Just because I stepped into the public discussion, and really, really bad stuff. But I didn't want to dwell on that too much. But the thing that was so good about this was how it woke up you in the scientific community. My boss wrote some great responses to this. Other folks said, we're not going to stand for this anymore. Scientific organizations said, you can't let this kind of fake news attack a scientist who's just sharing science. But the best response that happened came from folks from the skeptical community. And it was the Stephen Novellas, Joe Schwartz, David Gorski, Cara Santa Maria, Kevin Stanapathy, so many other people from the skeptical movement that said, they were the most vocal and said, you are not doing this to a scientist who's simply talking about science. And that was wonderful. But, on top of that, it was the podcasts folks. All the science podcasts stepped up and talked about it, gave me some time. But the biggest one was total strangers, people I never met who stood up and communicated in a very clear way that they weren't going to tolerate a scientist being attacked for speaking about science. And so that's kind of, the thing I need to leave you with, is how powerful it is for you as a science-minded community to step into these discussions, especially around issues like gene editing and genetic modification. Your audience is not moved by statistics. And I talked about that before. Sorry, Bill. Your audience is not moved by statistics. Your audience wants to know who you are, why you care, and and why they should trust you. You have to lead with your values. And when you're talking about climate, you're talking about vaccines, you're talking about genetic engineering, you're talking about why you should go to dinner somewhere else. Start the discussion with why you care, who you are, what's in your heart. Because the only way you're going to win that discussion isn't by giving more information. It's about sharing why you care. And it took me a long time to learn that as a scientist. I talk about the environment, farmers, consumers, and the needy. And those are the reasons I've stayed in academic science for my entire career and why I didn't want to work for a company. Because I wanted to make decisions about what I researched and research the areas that really I cared about the most. And so we have to start with, what are our values? Why do we care? What's important for us before we'll ever convince anybody about any science? So take part in the social media discussion. Practice transparency. Be nice. The one time that you're not nice and you go flying off the handle, someone will take a screenshot and it shows up on your university's president's desk, I hear. The other things that are important is that, kind of summing up the whole talk, is that humans have always changed plants and animals to better suit us, and that we've got an amazing safety record of genetic engineering and of food in general, and that fear and lack of trust really just impedes our progress at feeding more people healthier and safer food that's maybe more sustainable for the environment. Communication requires that trust, and don't ever, 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 ever go on this GMO versus organic thing. That I think, to me, we study organic stuff, organic production in my department, and I think genetically engineered plants for an organic production scenario is the perfect match. And I think that's where we'll be in 20 years. Talk about the strengths, but talk about the weaknesses. Don't be a cheerleader. Talk about the good and bad about technology. And so I probably should wrap up there, but the basic idea is I want you to understand that it's really just a small number of crops and a couple of traits. And that its most important thing is to think about the important solutions that have been impeded because of misunderstanding and miscommunication about these technologies. Share those with your family and friends. Learn about gene editing. It will change everything for us in agriculture and medicine if we let it. China has invested $4 billion into gene editing. That's almost our entire National Science Foundation budget, and that was before now. So we have some very important parts. Communicate with others, share the science, and do so with kindness, share your values. I will stop there, but if I can ever be of assistance, please reach out. Check out the podcast. It's a lot of fun. Thank you so much for listening, and we'll be happy to answer your questions.